Hello everyone, my name is Ludivine Sanchez-Arias, and today I will talk about the hydrodynamic effects of Mastigonemes distribution in Cryptomonad Flagella. I worked with Professor Henry Fu, and so in this video you can see a genus of Cryptomonads, it's called the Chillomanas Paramecium, swimming under optical microscopy. To illustrate my introduction, I'll play a small video that I think shows beautiful images. So Plankton is a diverse collection of organisms that live in a large body of water and whose powers of locomotion are not sufficient enough to prevent them from being passively transported by the currents. As you can see, it's a broad spectrum of organisms that includes bacteria, archaea, algae, protozoa, and other drifting or floating animals. And they are very important to study. So aside from representing the bottom few levels of the food chains that support our fisheries, this ecosystem plays a role in different cycles in ocean, different chemical cycles, and among them the ocean carbon cycle. Also, it's important to know that the phytoplankton, the photosynthetic part of the plankton, releases nearly half of the world's oxygen every year. And we believe that the mechanistic approach to plankton ecology can help untangle the processes among these organisms' growth and food webs, as well as understanding better the consequences of climate change on the microbial population in ocean. So this covers different topics, a different area of research, and we focus more on studying the mobility patterns and the propulsion systems. However, all of these topics are constrained by the physical and the chemical characteristics of the medium, so of a 3D fluid environment. So in our research work, we will study cryptomonads, which are unicellular eukaryotes that can be found both in marine and freshwater environments. And in this picture, you can see the genus of cryptomonad called Chillomanas Paramecium. You can see that it has a flattened and elliptical pedicel with two unequal flagella. So this is a longer one and a shorter one. And we come to study these organisms because they may be responsible for a large part of the primary carbon production, and because they are also important to study a peculiar food chain where the next predator doesn't necessarily eat the whole body structure, but can actually steal the organelles, so what confers the function to the organism, and steal this function. So for example, here the dinoflagellate will steal the pigments and then can acquire the function of photosynthesis. And it's interesting because both of these predators are harmful in their own way. So the ciliate here is involved in harmful algal blooms and red tides around the world, while this dinoflagellate can threat our shellfish aquaculture by producing toxin. However, we will focus on this genus, which is actually non-photosynthetic because it was easier to grow in the lab. So these cells are also interesting to study because they bear mastigonemes, which are rigid, thin nanometer scale filaments that project more or less normal to the flagella. The first images are from the 60s with EM imaging, and then their composition has been studied, so they are known to be made of a protein that is rigid like microtubules, and they are quite diverse. So within these groups they can be arranged in 2 rows or 1 row with or without terminal filaments, and yet their function remains unknown. They could be involved in photosensory, so orienting the cells towards the sunlight, or mechanosensory, helping the cell to feel the small perturbation in water, for example, like the filaments on a copepod antenna. Or they could be involved in mating, and finally they could help creating a feeding current, or they could affect the locomotion, and this is where our work will focus. We will focus on the hydrodynamic effects of the Mastigonemes. So this topic has been studied since the 60s with Holwell and Slate that investigated the flagellar motion in Locomotion, which is another protist, so in this case a golden algae. And they noticed that the wave motion and the translational movement of the organism were in the same direction. This is quite unusual because when we think of a traveling wave along a filament, it usually leads to the propulsion in the opposite direction. For example, if we have a base to tip traveling wave, in a flagellate organism, let's say sperm with a slender body, well, we can divide its flagellum into different segments, and when the wave is traveling along the segments, the material points will be pushed up or down, depending if they are in the red or the green sections. But the resulting drag of the flagellum implies the resulting motion of the whole organism. So here, if we have a perpendicular drag that is bigger than the tangential drag, then the material points of our segments will not just move up or down, but they will also slide forward with some angle, because in this case they want to swim in the tangential direction. And the net effect is that overall this whole organism will move this way, so in the opposite direction from the traveling wave. But so this was using this ratio, so less tangential drag than the normal drag. And Howell and Slay suggested that this could change if there is less normal drag than the tangential drag, in which case the segments want to move more in the normal direction, and ultimately, if there is enough tangential drag, then the net motion will be, in this case, backward, so in the direction of the traveling wave. And they derived an equation using the resistive force theory that describes the velocity to be proportional to 1 minus the ratio of this coefficient, so 1 minus tangential over normal drag coefficient. Meaning that if the CT is larger than the CN, then the direction of our swimmer reverses. So other people have been investigating what is this hydrodynamic effects of these Mastigonemes, starting with Brennan in 1974. He studied a theoretical model and showed that by adding these rigid filaments on the flagellum, so here 2 rows within the beating plane, well, it would give us the change of drag coefficient that we need, and concluded that the organism will swim in the direction of its flagellum. We would then have thrust reversal because of an increase of the tangential drag coefficient. And in 2011, Namdio et al. Studied the Mastigonemes through a model using the boundary element method, again resolving them in 2 rows within the beating plane. And this is their main results with the pressure field and the streamlines, and they concluded that when we increase the flexibility of the Mastigonemes, we can decrease the hydrodynamics effects. Finally, this year, a group from Delft University studied the Mastigonemes of the green algae Chlamydomonas, and the main work that shows their conclusion was mostly experimental, comparing when they have Mastigonemes, so within the beating plane and again displayed in 2 rows, with a mutant that doesn't bear Mastigonemes. They showed that the velocities and the forces look very similar, and that a model that did not incorporate these Mastigonemes could predict those forces in both cases, suggesting that the Mastigonemes have no hydrodynamic effects. However, they studied different kind of filaments, as the Mastigonemes of Chlamydomonas are known to be shorter and maybe more flexible than the Mastigonemes of Cryptomonads. So overall, the literature is still missing a study of other geometries and arrangements of Mastigonemes that would not be necessarily within the beating plane and arranged in 2 rows, because we know that in nature other geometries are possible. And so there is a gap in resolving in 3D these filaments. We first needed to find the geometry of the distributions of Mastigonemes in our swimmer Chillomanas. And to do so, we tried different kind of microscopy, starting with optical microscopy. So here we have a spore swimming. And you can see that even though we can see the flagella, we can't see these Mastigonemes, the small filaments, as the resolution wasn't high enough. Using SEM imaging, we could define the cell body geometry, as well as the length and the thickness of our flagella, but again, we couldn't spot the Mastigonemes. However, using the TEM and a protocol of negative staining, we were able to see them and study their geometry. So here we have the 2 flagella that can be found here, and here all these little dots that can be seen here are actually the anchors for the Mastigonemes. We can see that the Mastigonemes are displayed in 1 row on each flagellum, and they are all spaced in average by 240 nanometers. We then used these dimensions to describe the geometry of our swimmer in 3D, as well as using an equation for the flagella centerline from Nguyen papers that was studying another protist. And our goal here is to obtain the velocity of Chillomanas of our swimmer. To do so, we solved the Stokes equation in the low Reynolds number region, using the method of regularized Stokeslets and using rigid Mastigonemes. So here in this picture, each point from our discretization is actually a Stokesled. So we compared three cases, having no Mastigonemes, so smooth flagellas that are beating in the YZ plane, or having Mastigonemes that are resolved in the beating plane, so one row on each flagellum in the YZ plane, or having the Mastigonemes that would be perpendicular to this beating plane, so in the XZ plane. We then obtain the velocity in the Z direction for our three cases. So here the smooth flagella case has a negative velocity in the Z direction, so as expected it is swimming downward. The case of a swimmer having the Mastigonemes displayed in the beating plane seems to have a velocity that decreases until ultimately we would have thrust reversal. However, the average velocity is still negative here. And finally, having the Mastigonemes resolved in the plane perpendicular to the beating plane seems to increase the speed of our swimmer by increasing the perpendicular drag. We also wanted to try a simpler geometry with only one body cell, one flagellum, and this time two rows of Mastigonemes, in order to explore the effect of having the Mastigonemes distributed in other planes, so in 3D. In this plot we have the same distribution than earlier, and we have a new one, the 3D one, where the Mastigonemes are distributed in both the beating plane and the plane perpendicular to the beating plane. So we have this little cross everywhere. As a result, we have, as expected, the same velocities than earlier. The smooth flagellum configuration is swimming downward. The configuration having the Mastigonemes perpendicular to the beating plane is swimming faster downward. And the one having them resolved within the beating plane is actually leading to thrust reversal, with this time an average velocity that is positive. And as expected, the 3D geometry leads to having an average velocity which is in between, so between the smooth and having the Mastigonemes resolved within the beating plane. This is because the forces will compensate between the two planes. We then computed the ratio of the tangential drag coefficient over the normal drag coefficient that describes the direction of the velocity according to Hall-Williams-Slade. And from the resistive flow theory, if this ratio is equal to 1, then we will have a reverse of the thrust. We found a ratio of 0.62 for smooth flagellum and almost the same number when we are in a configuration where the Mastigonemes are displayed in the plane perpendicular to the beating plane. So we could explain that actually if we have an increase of the tangential drag with the same ratio of the perpendicular drag, then this ratio doesn't really change. However, we found higher numbers when the Mastigonemes are displayed within the beating plane. In this case, you remember we had a velocity in average that was negative for this case and became positive when we added a row of Mastigonemes. So for these two geometries, we would have an increase of the tangential drag that would be higher, bigger, than the increase from the perpendicular drag coefficient, leading to a higher number of this ratio. So we notice that for a number that is between 0.72 and 0.78, we would have the change of direction of our swimmer. However, based on our results, the ratio is not equal to 1, as it would be predicted, but it shows a trend. So it shows that the propulsion is in the same direction as the propagation of the flagellar wave when the effective ratio of the drag coefficient increases. In conclusion, we showed that the thrust reversal happens because of an increase of the tangential drag, and that only having the Mastigonemes resolved within the beating plane can reverse the motion. However, having them distributed in the plane perpendicular to this beating plane enhance the swimming of our Chillomanas. But other geometries can be found in nature. So here this is a golden algae with a very complex geometry of distribution of the Mastigonemes, and it would be interesting to study the hydrodynamics. It would be also interesting to study if the Mastigonemes can be actively controlled, so we would like to try cross-sectioning and see if the Mastigonemes can reach all the way to the axoneme. We would like also to implement flexible Mastigonemes and to study and model the kinematics of Chillomanas flagella to find an equation for their center lines. All of that would be to test their possible functions, so we already showed that they affect the locomotion, but could they be involved in creating a feeding current, in the sensing, or in mating? Thank you very much.